Hi scholars, so today I will review with you the first part of chapter 3, that is through page 54. You should make sure you have your book open while you are watching the video and also have your IB biology assessment statements. And you should make sure you take notes because I'm going to be checking both your notes from the book and the notes from the video tomorrow in class. So, this section is called the chemistry of life, which another way to say chemistry of life is biochemistry. So you need to know that there are certain elements that are found in all living things. They are carbon, hydrogen, nitrogen, and oxygen. So these are found in all living things, and then there's other elements that are found in biological systems that are important in specific molecules, but these are the general ones. The other ones you have to know are sulfur, calcium, iron, phosphorus, and sodium. And if you look on page 47, you'll see a list of the elements and their role in plants and their role in animals and their role in prokaryotes. And you should know at least one function for each. But we'll work on that. So, in the next section they talk about the structure of water. And last year, I hope that you discussed the structure of water. It has this kind of bent shape because it's lone pair electrons are pushing the electrons in the bond away so they're evenly spaced from each other. So it has this bent structure. Now, because of that bent structure, I'm going to redraw it here without the electrons there, without the lone pair electrons. This side, because of the lone pair electrons, ends up being very negative. So we have the symbol delta. So for partial, this is partially negative. So we should write that. Partially negative. This is the partially negative side of the molecule, which makes this side the partially positive side. Remember, this looks like an S, but then you fill in you keep going with the tail of the S. And it makes this delta symbol, the Greek symbol delta. So this side is partially positive. Now what this ends up happening is it affects the properties of water, and we will talk about that on the next slide. So let's talk about the properties of water. If you draw the water molecule again and see that if this side is delta minus and that side is delta plus, well, you can attract another water molecule with the O pointing towards the delta positive side of the other water molecule. And what this does, it forms a hydrogen bond. So these parallel lines in the middle represent the hydrogen bonds that occur between water molecules. So a hydrogen bond is attraction between hydrogen of one molecule and the oxygen, nitrogen, or fluorine of another molecule. And if you remember from your periodic table that oxygen, nitrogen, and fluorine are in the top right-hand corner, which mean they tend to have a negative charge or a partially negative charge because of the lone pair electrons. And this means that they're also in the top right-hand corner, which means that they are smaller. So small negative or partially negative atoms are going to be attracted to hydrogen atoms, which are partially charged. This ends up making the molecules stick together. So on a penny, when you dropped water on it, right, you saw that it kind of beat it up like that, which means that the water molecules sticking together. That's cohesion. So it's the stickiness that comes from that. It's a stickiness that you see in water molecules. So this relates to its thermal properties too. Because the water molecules stick together, it takes lots of energy to break them apart. So when you heat water up, you have to add a lot of energy in order to boil it, right? It takes a lot more energy to boil water than it does alcohol, for example, or gasoline. because you form a lot more hydrogen bonds in between water molecules and it holds them together. Same thing for cooling them down. It takes a long time for hot water to cool down, and hope to do that lab on Friday. Here, for cohesion uses, you should say stickiness to each other. And that's what we saw in the penny. And if you were to use a graduated cylinder and you were to pour water in, you would notice that the water molecules stick to the side, right? That's called the meniscus. And the meniscus is the water molecule sticking to the side. So sticking to the container is adhesion. And water is a really good solvent for other polar molecules. So what do I mean by polar? Well, if we draw the water molecule again, polar means, and this is a really, really big idea, a polar molecule is a molecule that has sides. So there's a negative side and a positive side to the molecule. A nonpolar molecule would be the same on both sides. Same charge on both sides. I'm running out of space here. Okay, so for example, carbon dioxide. If you did Lewis structures last year, you drew carbon dioxide like this, with its lone pair electrons on the oxygen. And you notice if you draw a line that divides carbon dioxide in half, like that. Sorry, I was a little bit off there. But if you draw it in half like that, right, you notice that both sides are the same. So that means carbon dioxide is a nonpolar molecule. But if I were to draw a line down the middle of water, you'll notice the two sides are different. So as long as you can draw a line and get them to be asymmetrical like this, the molecules would be polar. So water acts as a good solvent because it dissolves other things that are just like it. There's a rule in chemistry that you should have learned, and this is called like dissolves like. And what this means is that polar molecules dissolve other polar molecules. Nonpolar molecules dissolve other nonpolar molecules. This is why oil doesn't dissolve in water, and why water dissolves salt, because salt has charges. Na is plus, Cl is minus, and so the charges in water, the plus side helps dissolve the negative charges in chloride ion, and the negative side of the water dissolves the positive charges of the sodium ion. So in the next section, it talks about biological molecules. And an important idea that you have to know for this section is the idea of monomers and polymers. A monomer is one small piece, and when you put the small pieces together, when you put many of them together, you get a large chain of them. So that's what a polymer is. Poly means many. So here's this little outline of a little cutout man, right? And I'm sure you've done this like in elementary school, where you chain them together, right? And if you take these and you chain them together, so you notice in the example where you now have two, this is called a dimer. And then you copy it again. And now we have a trimer. Now you get enough of them together and you can just call it a polymer, right? So this is the main idea you have to understand from this section 3.2, which starts on page 49 and 50. So what are the four major biomolecules that you have to know? Carbohydrates, lipids, proteins, and nucleic acids. So carbohydrates have the general structure where they are usually ring structures with an oxygen in the ring. And then you can chain these together to make a polymer. A lipid has two parts, so these individual parts are called monosaccharides, and you put them together and they're called polysaccharides. Lipids have a glycerol, which is a 3-carbon structure, and then it has a longer chain called a fatty acid attached. Now you may not have learned this last year, but if you see a structure like this, where it looks like a zigzaggy line, each point on there is a carbon atom. It's just assumed that it's a carbon. And remember that carbon bonds 4 times. But you only see this carbon here bonds to that carbon and bonds to that carbon, so you only see 2 bonds. The other 2 bonds, if they're not shown, are going to be hydrogens. And if you see a double line like that one, oops, like right there, that would mean there's a double bond there. So when you see these, instead of writing C multiple times, all scientists do is they write they skip it and they just write a zigzag line. So lipids are another type of biomolecule, and they're made out of fatty acids and glycerol. So proteins have the general structure. They have an N-terminal side and a C-terminal side. They are all the same. On the C-terminal side, they have a carboxyl group on the end. On the N-terminal side, it's called an amino group. In the middle, all molecules look the same, except in the middle, there's an R group, which is your variable side chain. Variable side chain. And you can put these together. You put the N-terminal side with the C-terminal side of another molecule. So if I were to draw another amino acid, this side, the C-terminal side of this amino acid, bonds to the N-terminal side of the other amino acid to make a polymer of a protein. So these individuals are called amino acids. When you put them together, they're called proteins. Or they're called polypeptides, right? Individually, they're called monopeptides or monomers, and as polymers, they're called polypeptides or polymers, okay, or proteins. And nucleic acid has 3 parts, and we'll go over this more in the next section. But it has a nitrogenous base, it has a ribose sugar, and it has a phosphate group. And again, and the examples of nucleic acids you have to know are DNA and RNA. And this is reviewed on page 50. And you have to know the 4 major classes and what their subcomponents or monomers are. And you see it in the lower table on page 50. You have to know the examples of monosaccharides, disaccharides, and polysaccharides. Something you just have to memorize. So in this last section, it talks about condensation reactions versus hydrolysis reactions. So you should think of, in your life, what condensation refers to. It refers to water, and hydrolysis refers to adding water. So this is water coming out, water leaving, and this is adding water to break up something. So in this case, you see that there's 2 monosaccharides that are attached to each other in sucrose. You add water and it breaks them up. So notice here there's an H, and when you add water, the OH from water comes to here, and the other H from water goes to here, and now you have 2 monosaccharides. So this would be called a hydrolysis reaction. because you're breaking up a larger molecule to make a smaller one. Now imagine you had an arrow going this way, where you're going to put two monosaccharides together to make a polysaccharide. Well, that would be called a condensation reaction, because you see, if you go from right to left, the water now becomes a product in this reaction going from right to left. So, and you see that the water, where does the water come from? Well, it comes from this OH, and from that H, you put them together and you get H2O, and the bond comes from this O and connects down there. And if you look at the examples that are on page 53 and 54, you will notice some other examples of going back and forth between hydrolysis and condensation. So please make sure that you watch this video again so that you can take the notes and understand it to the best of your ability. I will see you in class tomorrow. Bye.